Hi everybody. Let us continue with the chapter Sexual Reproduction in Flowering Plants. So in the previous video we have discussed about the structure of micro and megasporangium and how are the male and female gametes are formed in the micro and megasporangium respectively by the process of microsporogenesis and megasporogenesis. Now once the gametes are formed, the next step is the meeting of gametes. To make that happen or to make the gametes meet each other, pollination is the process. So let's talk about pollination. Now, what is pollination? Pollination is the transfer of pollen grains from anther to stigma. When the pollen grains are transferred from anther, so this is anther, to stigma, which is the landing zone, it can either happen within the same flower or to a different flower of the same plant or a different flower of a different plant of the same species. Based on the source of pollen grain, pollination can be widely classified into two types: self-pollination and cross-pollination. Self-pollination can again be categorized into two: autogamy and geitonogamy. Autogamy is the transfer of pollen grains from anther to stigma within the same flower. It happens within a flower, whereas geitonogamy is the transfer of pollen grains from anther to stigma of another flower but within the same plant. Though it is happening to a different plant, it is a genetically similar plant. That is why it can also be said as a self-pollination. Whereas cross-pollination, that is xenogamy, the pollen grains are transferred from anther to stigma of totally different flower which is in another plant but of the same species. It is very important that it should be of the same species. If you closely observe, you can see the self-pollination does not require any agents to carry out, whereas geitonogamy and xenogamy require the pollinators or the pollinating agents. In some plants, there are two kinds of flowers. One above the ground, that is chasmogamous flowers, and the other one below the ground, cleistogamous flowers. Chasmogamous flowers are quite open and normal like the other flowers, where the self and cross-pollination is possible. Whereas the cleistogamous flowers are closed so that the anther and stigma are enclosed within that. So there is no chance of cross-pollination. Either geitonogamy or xenogamy is not possible in the case of cleistogamous flowers because they are closed flowers. So in this case, purely self-pollination is possible. You can see the chasmogamous flowers and cleistogamous flowers in some species like commonlina and viola plant. The one which are found above the ground, they are open, and the one which is found below, they are closed, where only autogamy is possible. As we mentioned earlier, for both xenogamy and geitonogamy, pollinators are very, very essential. They play a major role because without the help of pollinators, the pollen grains cannot be transferred from anther to stigma in case of xenogamy or geitonogamy. In plants, both biotic and abiotic agents act as pollinators. Let us discuss about the agents of pollination or pollinators. Abiotic agents can be either wind or water, and biotic agents can be insects, animals, which includes everything. It can be bird or an animal or a human being, any kind of living organisms. If they played a major role as a pollinator, then they fall under the biotic agents. And now let's discuss about the characteristics or uniqueness of flowers pollinated by these different agents. Let's start with the abiotic pollinators. Pollination by wind, which is otherwise termed as anemophily, it is considered as more common among all the abiotic pollinators. Flowers pollinated by wind will mostly have pollen grains which are light and non-sticky. The pollen grains remain light and non-sticky so that it can be easily carried away by the wind. They often possess well-exposed stamens. In this flower, you can see the stamens are well exposed so that the pollen grains can be easily carried away by the wind. Also, you can see the stigma here is feathery so that it can easily trap the pollen grains carried away by the wind. Mostly the anemophilous flowers will have one ovule within each ovary and many flowers packed into an inflorescence. And what you see here is a tassel of a corn cob, and this tassel is nothing but the combination of style and stigma, which wave in the wind to trap the pollen grains. So the pollen grains can be easily trapped by them. Most of the wind pollinated flowers are very small, not that conspicuous in size. They do not have special fragrance or colors so that it cannot attract the other pollinators like biotic pollinators like insects or other animals. And now since they have lightweight pollen grains, they can be easily carried away. So this is the flower of grass. Mostly in grasses, wind pollination is possible because the flowers are very small and the pollen grains are more in number and light in weight. Now let's talk about the pollination by water, which is otherwise called as hydrophily. Water pollination is mostly seen in the plants which live near the water or in the water bodies. It is mostly found in monocotyledons. But if you just check the other group of plants, lower group of plants like algae, bryophytes, etc., hydrophily is seen there. In the case of angiosperms, that is the flowering plants, hydrophily is found in most of the aquatic plants, but not in all aquatic plants. In some of the aquatic plants like water lily and water hyacinth, the pollen grains are exposed, the flowers are open above the surface of the water. The flowers of these plants are beautifully colored so that it can attract the biotic pollinators like insects and birds. And the pollination is conducted by the biotic pollinators in these plants where the flowers are open and are found above the water. But in certain plants where the flowers are found submerged, only hydrophily, that is water pollination, is possible. Some of the water plants like Vallisneria, you can see here the female flowers are seen above the water surface. The male flower releases the pollen grain, or sometimes the male flower itself will be opening out, separated out from the inflorescence, and it will be moving towards the female flower passively according to the or along with the water current, reaches the female flower and it gets pollinated. Another example like seagrass, the whole plant is submerged under the water, and here the pollen grains are released inside the water. They will be taken passively along the water current, reaches the female flower, the stigma of the female flower, and achieve pollination. And in these cases, water acts as the agent of pollination. And in both, one of the unique feature of the pollen grains of this water pollinated flowers are they have a mucilaginous covering which protects them from getting wet by the water. And remember, both the wind and water pollinated flowers do not produce nectar. That is one of the characteristic feature of both wind pollinated and water pollinated flowers. Also, do you see any of these flowers colorful and conspicuous, large in size? No, they are not colorful and they do not produce nectar. Can you guess what is the reason for that? Because they need not attract the biotic agents for pollination. The pollination is carried out passively by either wind or by water. So color or the formation of nectar doesn't play a major role here. When the pollination is carried out by the living organisms or the biotic agents, it is generally called as zoophily. Zoophily can be again categorized like entomophily. Pollination is carried out by the insects. Ornithophily, the pollinators are birds. Chiropterophily, where the pollinators are by bats. And malacophily is conducted by snails. Now, among this, insect pollination is more common. Among the insects, bees are the most commonly found pollinators. Other than this, even larger animals like rodents, some kind of reptiles, and even lemurs are also found carrying out the pollination. Animal pollinated flowers are mostly species specific. That is, those flowers will be having special characteristics which support these animals to pollinate that flowers. For example, if you just check the insect pollinated flowers, most of the insect pollinated flowers they'll be large, colorful with fragrance, and they'll also be rich in nectar. If the flowers are small, then those flowers will together form an inflorescence, and so that those flowers will be looking conspicuous or big. So group of small flowers which are called as inflorescence, so they appears to be a big one. Bees or other insects are attracted to these flowers by their conspicuous size, their colors, fragrance, or sometimes even for nectar. They visit to a plant, and while sucking the nectar, the pollen grains will get attached to their body, and once they visit another flower, these pollen grains are shed to the stigma of the other flower. That's how these pollinators carry out the pollination. To attract the insects again and again, or to sustain their visits, these flowers will provide certain rewards to these animals. So the rewards can be like nectar, or sometimes pollen grains can act as a reward. To harvest the rewards, or to get this reward again and again, these pollinators visit the flowers. In some species like Amorphophallus, which is the tallest flower, this flower is more than 6 feet in height, and in this flower the floral reward is like they'll provide safe places for the insect to lay eggs. So these flowers usually produce a foul smell, and those foul smell attract the insects for pollination. And since the surface area of this plant is huge, insects get a safe place to lay their eggs, and so that the eggs can hatch out without the threat of other predators. Another interesting floral reward is given by the yucca plant to a moth called yucca moth. So the relationship between yucca plant and the yucca moth is very interesting, such a way that this plant can be pollinated only by this moth. And the plant provides safe place for this moth to lay eggs, and so that their larva will grow safely within this plant, feeding on their seeds. To know more about the relationship between the yucca plant and yucca moth, you can watch the video. The link is provided in the description box. Majority of the flowering plants produce hermaphrodite flowers. So what are hermaphrodite flowers? The flowers that possess both stamen and pistil within it. Both stamen and pistil within a flower are called hermaphrodite flowers. So hermaphrodite flowers mostly give a chance for self-pollination. So when this self-pollination is continued to happen, it will lead to only genetically similar seeds. So to avoid this condition, nature itself has provided certain outbreeding devices, and those outbreeding devices would control the occurrence of self-pollination, and so that it enhances cross-pollination, the chances of cross-pollination, so that the chances of genetically dissimilar plants are more, which leads to variation. So some of the outbreeding devices explored or identified are dichogamy, heterostyle, self-sterility or self-incompatibility, unisexuality, and diacy. Let's see these conditions one by one. Dichogamy is a condition where the pollen or the stigma receptivity are not synchronized. It means either pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. This condition can also be classified or identified as protandry and protogyny. Protandry is the condition where the stamens get matured earlier before the stigma becomes receptive. So you can see the anther is dehiscing and the pollen grains are released out while the stigma is still underdeveloped. Once the stigma becomes receptive, there are no pollen in the same flower. So protandry is the condition where the androecium or the stamen is matured early before the stigma becomes receptive. Protogyny is a condition on the other way around where the stigma becomes receptive earlier before the stamens gets matured. Next condition is identified as heterostyle, where you can see the positions of anther and stigma are different. In this both flowers, you can just find the difference in the position of stigma and the stamen. Here the stigma is over here, the stamens are here, so that the pollen cannot come in contact with the stigma of the same flower. So the chances are very less where the stamens of this flower falls on the stigma of the same flower. So this is also a condition to avoid self-pollination, which is an outbreeding device, that is nature enhances the chances of cross-pollination. Self-sterility or self-incompatibility is a condition where the pollen grains from the same plant is not accepted by the stigma of the flower. You can see the stigma of the flowers is receiving the pollen grain. So pollen grain S1 and S2 is not accepted by the stigma of this flower, and in this condition you can just see the stigma is not accepting S1 pollen grain, and here the pollen grain from a different plant is being accepted by the stigma. This is a genetic mechanism. This mechanism prevents the pollen from the same flower fertilizing the ovules, and the germination of the pollen tube is inhibited by this pistil. So this condition is called as self-incompatibility or self-sterility. Another outbreeding device to prevent the self-pollination is the formation of unisexual flower. So in this flower you can see this is a female flower with only the gynoecium in it, and this is a male flower with only the stamens. So obviously this stamens or the pollens from this stamen have to pollinate another flower. So self-pollination is totally inhibited here. In this case, if both male and female flowers are present in the same plant, we call it as monoecious. And if the male flower and the female flower are found in two different plants, we call the condition as dioecious. So dioecious plants possess separate male plant and a female plant, which has only the male flower in it and only the female flower in it. If the condition is monoecious, then there is a chance of geitonogamy, that is, the flowers of the same plant can be pollinated, but in the case of dioecious, strictly xenogamy only will be occurring. And dioecious condition will prevent autogamy and geitonogamy. So autogamy or geitonogamy is not possible here, and in this case autogamy is not possible, but there is a chance of geitonogamy. Okay. Once the pollination is completed, it is not guaranteed that the fertilization or the germination of pollen tube will happen. We have earlier discussed about the self-sterility or self-incompatibility, where the correct type of pollen grains only can germinate on the stigma. The wrong type cannot germinate on the stigma, though it lands on a stigma. Now, this compatibility is being recognized by the pistil. So, this recognition is done by pistil. The pistil has the ability to recognize which is the right pollen to germinate or which is the compatible one, and only that pollen grain will be allowed to undergo the post-pollination events. The post-pollination events are nothing but fertilization. If the pollen which is landed on the stigma is of the wrong type, the pistil will reject it and the pollen grain cannot germinate. That is, no pollen tube will be formed there. Now, this interaction between the pollen grains and the pistil is mediated by certain chemicals, and this interaction is termed as pollen-pistil interaction. Once the pollen and the pistil interacts, they find recognizable or compatible each other, then the post-pollination events start occurring. So, the pollen-pistil interaction involves pollen recognition followed by promotion or inhibition of the pollen. If we desire to create a commercially superior variety, artificial hybridization is one of the major methods. See, in artificial hybridization, we can pollinate it artificially. While conducting this artificial pollination experiments, the pollen grains, that is the desired pollen grains which we use for pollination, as well as the stigma which receives the pollination, has to be protected from contamination. It means if we select a plant with desirable character as a female flower, we should be very careful that the stigma of that flower should not be contaminated with the pollen of any other different plant which we have not selected. In that case, we can cover the female flower by a method called bagging. Also, we can conduct emasculation. Emasculation is a process where the stamens are removed artificially or manually. We could remove the stamen so that the stigma shall not be contaminated by the pollen grains which we do not desire or which we have not selected. When the stigma of this flower which we have bagged has reached the stage of receptivity, the pollens which we have collected from the desired anther can be powdered on the stigma. And again these flowers are rebagged and are allowed to undergo fertilization, and the fruits are allowed to develop. So this method is called as artificial hybridization. This is one of the method of artificial hybridization. In case if the female plants are unisexual flowers, then there is no need of emasculation. Emasculation is not necessary. We conduct only bagging there so that we can protect the stigma being contaminated by the pollens from the undesirable plants. Pollination is followed by the germination of the pollen tube. So you can imagine that this is the stigmatic surface where the pollen falls, and once the pollen is identified or recognized to be compatible, it starts germinating, and you can see the pollen tube over here, the vegetative cell and the generative cell. The vegetative cell with this vegetative nuclei is otherwise called as the tube nucleus, which leads the way. Generative nuclei splits to form two male gametes, whereas the embryo sac is ready with the antipodals at the chalazal end, the central cell with two polar nuclei, and the micropylar end with two synergids and an egg. You can find the filiform apparatus in the synergids, which helps in the reception of the male gametes and leads their way towards the female gamete. So you can see the pollen tube is germinated. The pollen tube reaches the micropylar end and it reaches near the synergids. And meanwhile, the pollen tube with the two male gametes reaches the micropylar end, And once it reaches the synergids, one of the male gamete passes through the synergids and fuses with the egg cell to form the diploid zygote. So one of the male gamete fuses with the egg cell to form diploid zygote, whereas the next male gamete fuses with the central nuclei to form the primary endosperm nucleus. So another male gamete fuses with the central nuclei, that is the polar nuclei. Polar nuclei are 2 in number, so N plus N. Another male gamete, which is of N in number, fuses to form the primary endosperm nucleus, which is 3N in number. Since there is a fusion of 3 haploid nuclei, we call this fusion as triple fusion. And this process is called as syngamy. Since twice the fusion occur, we call the whole condition as double fertilization. After double fertilization, the zygote develops into embryo and the primary endosperm nucleus will develop into primary endosperm cell, which ultimately develops to form the endosperm, which is triploid in condition, and this endosperm provides nourishment for the developing embryo.